So I hope I'm audible.
Yes, sir. Sir, I have met you the co-host, sir. You can share the screen and start the class, sir.
Fine. Okay. Thank you. Shall I start, right? Audio visuals are perfectly fine, right? Okay.
Yes,
Perfect.
sir. Yes, sir.
Okay. So yes, good evening, everyone. This side Dr. Abhijeet, and welcome to yet another session where we are going to start the basic chemistry today. That is the biochemistry of carbohydrates, part one. So, guys, you already know about the Oracle app, and we have initiated a 90 days class program, okay, where we will be revising the important questions, all the potent questions that are being asked in the upcoming or which are potent for the upcoming university exams, right? So, yes, apart from that, we are also covering everything relevant to the next exam as well, right? Whether it is next or whether it is your prof exam, I have always told you that common questions are asked commonly, right? So nothing changes. The important topic remains the important only. They will not change with the name of the examination if it changes. So yes, guys, today I will be discussing the format. I mean the questions, the topic in three different questions. The long essays, which are like 10 marks. The short essays, which are like 8 or 5 marks, right? And the short answers like 3 or 2 marks, right? So in totality, I will be discussing 8 questions today. I will be starting with the long essay one, then I will be covering the short essay questions, and then I will be discussing the short answers, right? Now, guys, why we choose Oracle? You already know we have the recorded classes. The ones who are not able to know, like attend the live class, they can always go on for the recorded classes that are available on the app. The live classes, we are covering everything live on YouTube, right? Every important topic, every important question is being covered live on YouTube. All the practical sessions I will be discussing after completing this 90-day class, right? Even the practical sessions are being recorded, or it's like uploaded on the app as well. We have the exam module, right? And apart from that, the very important thing that is now available with the Oracle app, it is the hard copy book set. So wherever in whichever corner of the India you are, you can just order these book sets, and these notes are basically curated in the form of Q and A. That is the question and answer format. That is something very much essential for the examination point of view. So we have the recorded classes, we have the live classes, guys. We have the express exam module. I have already told you, which we are covering like 10 years of question papers, right? Apart from that, one-to-one doubt clearing session is also being provided, right? You can just book. And the hard copy notes, it is also like provided, right? We have the question bank, guys. I have already told you it's from basic to advanced level, right? Basic to advanced level. The clinical questions are also being covered over here, right? So whether it is next exam also, where it's more like clinically oriented nowadays. So obviously we have some basic questions, one-liner questions, but beyond that, we have the clinical questions also, which are designed by the expert faculties, right? In keeping in view about the upcoming next pattern examination, right? We have the practical sessions, as I have already told you, it's being recorded on the app, right? So this is the basic subscription plan, a 3-month plan, 6-month plan, right? 12-month plan, and a 4-year plan, right? For additional discounts, guys, you can just use my code Dr. Abhijeet 20. Now let us start with the very first question. Explain the classification, structure, and biological significance of carbohydrates. So we have started with a 10 marks question right now. We have started with a 10 marks question right now. It's a long essay type of a question in like the MBBS University Prof 1 examination. So the very first question, you have to first understand, guys, what is carbohydrate. Firstly, Additionally, you should know over here what is carbohydrate. What is carbohydrate? Now, the basic definition, guys, carbohydrates are nothing but hydrates of carbon, right? So the basic definition says carbohydrates are nothing but hydrates of carbon. So it contains carbon, hydrogen, oxygen, right? So these are hydrates or carbon of carbon. And other important definition that you have to remember from the exam point of view, and that will again help in understanding the structure of the carbohydrates. So carbohydrates are nothing but carbohydrates are aldehyde. Now, when I say aldehyde, you guys already know the functional group. It is CHO, C double bond O H, right? This you can also write it as CHO or keto derivative. Now, when I say keto, guys, you already know it is C double bond O, right? Keto derivative of polyhydroxy alcohol. Polyhydroxy alcohol. Now, guys, let us just try to understand this definition. Now, I have told you this is polyhydroxy alcohol. Poly means many, hydroxy means OH, and we have alcohol. Now, for this context, I'm taking an example over here. I'm taking 3 carbon, right? I am taking 3 carbon, guys. You already know, right? Hydroxy means OH group, right? To all the carbons, I'm adding OH group. And what is the valency of carbon, guys? You already know it is 4. It is 4. So basically, to balance this carbon, basically we have to make 4 bonds. So I will be adding 2 hydrogen over here. I will be adding 1 hydrogen over here, and I will be adding 2 hydrogen over here. So you just numbering 1, 2, and 3. You can see that over here. This is basically the structure of glycerol. It is the structure of glycerol, and it is nothing but polyhydroxy alcohol. Now, I have told you in the definition that carbohydrates are aldehyde or keto derivatives of polyhydroxy alcohol. Now, from this polyhydroxy alcohol, I have to make aldehyde or keto, right? Now, guys, firstly, I will be trying to make the aldehyde part over here. So that is basically the structure also, guys. The basic structure you should know over here. Now, aldehyde. How will I make aldehyde from the first carbon, guys? You already know I will be removing an H2. Okay, I will be removing 2 hydrogen, guys, over here, right? One from this one from here, and one from this. 2 hydrogen if I remove. So you can just see that the first carbon will be available with only oxygen, right? And out of this 2 hydrogen, only one will be left. So we have 1 H, rest the structure remains the same, right? So obviously for the carbon to have 4 bonds, it will be bonding with oxygen with 2 double bonds. So that is an aldehyde group. And to make an aldehyde group, we are removing hydrogen from the first carbon, right? So that is actually the structure which is called over here, that is called glyceraldehyde. It is formed from glycerol, and we are adding the aldehyde group over here. So this structure, it is called as glyceraldehyde, right? The structure is called as glyceraldehyde, right? Always remember, guys, that in an aldehyde derivative, the group is always present at first position. We have 3 carbons, it is always present at the first carbon. That is something you have to take over here. It is always present at first carbon, right? Now, one thing I have justified from the definition, that is an aldehyde derivative or keto. Now, to make a keto derivative, guys, to make a keto derivative, let me use another pen over here. The keto derivative, I have to remove, yes, I will be removing 2 hydrogens from the second carbon. Obviously, if 2 hydrogens are being removed from the second carbon, Okay. The first will remain the same. The second, only oxygen will be left with the carbon. And just to fulfill the valency of the carbon, will make double bond O, right? Because two hydrogens are already removed. And we have the third carbon. Yeah. So this structure, guys, obviously you are very well aware that if I'm just highlighting this particular portion, this structure is of acetone. CH2, C double bond O, CH2, it is called acetone. So obviously, and we are having two hydroxy group. So the name goes dihydroxy acetone. First position, OH group. Third position, OH group. Two positions where OH group is present, dihydroxy, and the yellow color shaded portion is acetone. So basically, you can see the simplest carbohydrates that are formed, glyceraldehyde and dihydroxyacetone, right? So that is something you have to remember. The two simplest carbohydrates, very, very important. The two simplest carbohydrates, right? That is glyceraldehyde and dihydroxyacetone, right? If the question says the parent carbohydrate is, it is glyceraldehyde only. Parent alcohol is glycerol. Parent alcohol is glycerol. So that is the basic structure. Firstly, you have to just remember of the no carbohydrates, right? So the simplest carbohydrates are nothing but the glyceraldehyde and the dihydroxyacetone. Now, coming on to the classification of carbohydrates. Coming on to the classification of carbohydrates. Now, guys, you can very well see. Now, the classification of carbohydrates, guys, always in the exam, try to draw a flowchart for the question. I've always told you, no one is going to read your no stories in the questions, right? It is always advisable to write the answer in the form of flowcharts, diagrams, and points. And that is something by no keeping this particular point in view. The carbohydrates, or let me just try to summarize in my own way over here. Yes, the carbohydrates classification. So the carbohydrates are divided into, yes, you already know. We have the monosaccharides, we have disaccharides, right? We have oligosaccharides, and the last one that is the polysaccharides. Polysaccharides, right? Monosaccharides, guys, you already know. When I say mono means one. Mono means one. So over here we have one sugar unit, one potential sugar unit. Disaccharides means two potential sugar units are there. Oligosaccharides 3 to 10, or I can say few sugar units are there. And polysaccharides means more than 10 sugar units are there. More than 10 sugar units are there. So that is the basic classification that you have to remember. Okay. Although monosaccharides and disaccharides I will be discussing in detail in the upcoming question. But as of now, monosaccharides, guys, you have to again remember the example over here. I will be discussing this, but as of now, remember monosaccharides example, we have glucose, galactose. Yes, we have glyceraldehyde. We have just drawn it. We have dihydroxyacetone, right? So these are various examples that you have to remember. Coming on to disaccharides, guys, you already know we have reducing and non-reducing. I will be coming to the terms reducing and non-reducing in a while. But as of now, guys, you have to remember the reducing ones are, yes, what is meant by reducing and non-reducing, I will be teaching you. The reducing ones are maltose, isomaltose, lactose, and lactulose. The non-reducing ones, guys, sucrose, right? Trehalose. So these are the non-reducing ones. Now, coming on to the oligosaccharides, they have classified disaccharides under oligo. Obviously, we can do that. We have trisaccharides containing 3 potential sugar units, raffinose, and tetrasaccharides, it is stachyose, right? Stachyose. Coming on to polysaccharides, guys, you have to just remember the homopolysaccharides and the heteropolysaccharides. The names as of now. I will be discussing this in detail. As of now, just try to remember the basic classification, guys. The ones containing like more than 10 potential sugar units. You should remember the homopolysaccharides are starch, dextrin, glycogen, cellulose, inulin. The heteropolysaccharides are hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate, keratin sulfate, right? I will be discussing this in detail. So that is like the classification of the carbohydrates that you have to just remember from over here, right? The classification of the carbohydrates. Structure, guys, for the monosaccharides have already told you. Okay. Now, if we talk about the structure of the carbohydrates, structure of carbohydrates. Now, guys, so basically you already know the carbohydrates are mainly composed of carbon, hydrogen, and oxygen. We have various structures of carbohydrates. For example, if we talk about monosaccharides. So monosaccharides can exist in the linear form also. Okay, just like I have shown in the very first part, the first slide, it can exist in the linear form, the glyceraldehyde and the dihydroxyacetone, even the glucose, galactose, they all exist in the linear form, right? So yes, we have the linear form and we have, yes, the cyclic form also. We have the cyclic form also, right? Or I can simply say that the ring form, right? Or the ring structure that is like very common in aqueous solutions. It is common in aqueous solutions, right? Secondly, we have the disaccharides. Now, you already know disaccharides are the ones where two monosaccharides are linked together by a glycosidic bond. Okay. So basically, we can just involve the two cyclic structures, which are combined together by the help of a loss of a water molecule. And hence how the disaccharides are formed. We'll be discussing it in detail as well. We have polysaccharides, guys. Polysaccharides, they can be either branched or they can be unbranched as well. They can be unbranched as well, right? For example, if we talk about starch, okay, starch has both amylose and amylopectin, right? Amylose and amylopectin. Now, guys, remember that amylose is basically a linear structure. Okay, it's a linear structure. Amylopectin, it is a branched. It is a branch structure, right? It is a branch structure like this. So that is something that you have to remember over here. Okay. Or cellulose. We can also talk about cellulose, guys, over here. That cellulose is also something which is having a linear or a straight chain structure, linear chain structure. Okay. So yes, that is basically the different structures of the carbohydrates that was as of now you have to remember, right? And coming on to the last one, that is the biological significance of the carbohydrates. Now, the biological significance. Now, guys, firstly, whenever you think about carbohydrates, it is always an energy source, right? You already know we are actually consuming the disaccharides. Disaccharides is going into no going into the stomach, converting into like monosaccharides. So obviously the energy source they are coming from the monosaccharides, the glucose, right? Monosaccharides like glucose, they are basically the primary energy sources for cell, right? Disaccharides, basically they are going inside, they're broken down into monosaccharides, and ultimately the energy is released, right? Apart from that, they are also, if energy is released, energy is stored also. Yes, energy storage. Now, when I say energy storage, guys, you already know the polysaccharides such as glycogen. We have already read in +1, +2 that the storage form of glucose or storage form of carbohydrates in animals, it is in the form of glycogen, right? And starch, basically, it is in plants. So that is something you already know. So it can be no, the energy is reserved, and whenever the body requires energy, this can be like broken down. The glycogen breaks down into glucose, and ultimately the energy is being obtained from there. These are the structural components, guys. When I say structural components, I've already talked about a term called as cellulose. Now, cellulose provides structural support in plant cells, right? Um, Plant cell, even if I have already taken a non term called as chitin. Chitin basically serves as the similar function of support in the exoskeletons. Exo means bahar, means outside. Exoskeletons of arthropods, or most commonly, it is the fungal cell wall. Fungal cell wall, right? Apart from that, you can also remember they are very, very important in cell signaling and recognition. They are very, very important in cell signaling and recognition. Now, we have already drawn a cell, right? A cell. Cell is basically covered by cell membrane or plasma membrane. Now, the plasma membrane, on the surface of the plasma membrane, we have the carbohydrates which are present. Carbohydrates which are present. So basically, these carbohydrates are nothing but the glycoproteins or glycolipids, right? And basically, they are crucial. They are very, very important for cell recognition. Okay, signaling process. Okay, they also impact our immune system, cell communication, right? So that is again you can remember. Apart from that, if you want to remember one more from the exam point of view, that they are involved in various biological processes. So you already know that the carbohydrates are involved in various biochemical reactions that are taking place in the body, for no, even in the synthesis of the nucleotides, the amino acids, other molecules also. Basically, it's like very, very important. So these are the biological significance that we have discussed, right? These are very, very important that you can just mention from the exam point of view. This is the biological significance. We have also discussed about the structure, guys. We'll be coming on to in detail about the structures right now. I will be drawing for you people. So as of now, no need to worry. The classification is very, very clear. The mono, di, oligo, and polysaccharides, and ultimately their examples are very, very clear. And we have discussed the basic definition and how the carbohydrates are being derived, like the structures, the basic structure we have already discussed in detail. So that was regarding the first question that you have to like pay attention from the exam point of view. Now, beta, coming on to the second question, that point, which is our point of discussion for today, that is the, yes, the 10 marks question. Describe the structure, properties, and functions of monosaccharides and disaccharides. Monosaccharides and disaccharides. Now, firstly, I will be discussing about the monosaccharides. Now, when I say monosaccharides, guys, please try to remember important things about that. It is the simplest form of carbohydrates. Okay, it is the simplest form of carbohydrates, right? So they are the simplest form of carbohydrates. Basically, they are consisting of like single sugar molecules, right? Now, general formula that you have to remember, very, very important for the structure. You have to remember the general formula. General formula, and that is nothing but Cn, and we already know we have H2n, right? Where n basically depicts the number of carbon atoms. Number of carbon atoms, and over here the carbon atom should be, or n should be, like more than 3. N or the carbon atom should be more than equal to 3. Okay, for example, we have already seen the simplest one, guys. The simplest one, we have taken 3 carbons, right? We have taken 3 carbons, 1, 2, 3. So we have glyceraldehyde and dihydroxyacetone, right? So that is if we talk about, okay, let me just try to make a table for you people. You already know. So we have, depending on the number of carbon atoms, depending upon number of carbon atoms, we have like 3, minimum 3, 4, 5, 6, right? 3 basically it is called as triosis. 4 it is called as tetrosis. 5 it is called as pentosis. And 6 it is called as hexosis. Right? Triosis, you already know. Basically, I've already told you these carbohydrates are aldehyde. So aldehyde derivative is called as aldosis, and keto derivative it is called as ketosis. Aldehyde or keto derivative. So the aldehyde group, if it is there, right, CHO, and keto group, C double bond O, that is ketosis. For 3 carbon atoms, we have already know the aldehyde derivative. It is glyceraldehyde. Keto derivative, you already know. We have dihydroxyacetone. We have just drawn on the very first slide. 4, it is erythrose, and ketosis, it is erythrulose, right? 5, we have like 3, the ribose, xylose, arabinose. Over here, for ribose, we have ribulose, right? Xylose, we have xylulose, and for arabinose, we have nothing. 6, you already know glucose, galactose, mannose, okay? And for this, we have only fructose with a 6 carbon atom, right? And the keto derivative, it is present at the second carbon. So that is basically the different like the basic structure where you have to remember on depending on the number of carbon atoms how these can be formed, and the structure of the third, the triosis, we have already discussed in the very first slide, right? We have already discussed this glyceraldehyde and dihydroxyacetone. Okay, so yes. So you can very well remember, guys, over here, the glyceraldehyde. Now, they have asked what is the biochemical importance, the functions also of these monosaccharides, right? So yes, firstly, I will be discussing the functions, then I will be coming on to the properties also of monosaccharides. So basically, glyceraldehyde 3-phosphate is an intermediate in glycolysis. Now, guys, you have already discussed about glycolysis in +1, where the glucose is broken down to form pyruvate, right? Where the glucose is broken down to form pyruvate. And in that, basically an intermediate is formed that is called as no glyceraldehyde 3. There is a series of reaction, and in between, basically an intermediate product you will just get over there, that is glyceraldehyde 3-phosphate, right? Erythrose, basically, it is an intermediate in carbohydrate metabolism. All the metabolism we'll be discussing in detail. The ribose, guys, especially, it is like it for the structure of RNA, for the structure of adenosine triphosphate, NAD, NADP. For the structure of DNA, deoxyribose is required, right? So these are all very, very important, guys, right? So yes, okay. So you just have to remember all these, right? And if we talk about the properties of monosaccharides, now coming on to the properties of monosaccharides, even you can also remember, guys, like this portion I have taken from the Auricle notes, and they are very well designed over here, which I can say. So glucose, galactose, mannose, you already know glucose, it is the sugar fuel, okay, of our body. It is the galactose, it is converted to glucose. Failure leads to galactosemia, a condition which we'll be discussing in detail, right? The heptosis, basically, it is an intermediate in HMP pathway. I will be discussing this hexose monophosphate shunt, okay, which is a major pathway for the production of NADPH, which is required for all these RNA compounds and everything. So, yes. So these are basically the biological importance, or I can simply say the functions of monosaccharides, the functions, right? Apart from that, if we talk about the properties of monosaccharides, properties of monosaccharides. So guys, remember the very first one, it is the solubility. So basically, they are soluble in water. They are soluble in water due to their ability to form the hydrogen bonds. Okay, they can form the hydrogen bonds, and that is one of the property of the monosaccharides. Apart from that, you have to remember that they are reducing sugars. They are reducing sugars, right? So now, when I say reducing sugars, guys, right? Now, I am actually talking about that they have one free functional group. They have one free functional group which they can donate anytime, guys. They can donate anytime to other molecules which are participating in redox reaction. So remember, guys, they are reducing, right? And apart from that, they also have a very sweet taste. So they are basically the glucose and the fructose, right? So they tend to have a sweet taste. So basically, we are consuming it. Okay. So that is sweetness is also a property of the monosaccharides apart from solubility and reducing property. So reducing, you already know they are reducing sugars. They can donate electrons to other molecules which are participating in the redox reaction. Now, coming on to the disaccharides, guys, coming on to the disaccharides. So the question has two parts, guys. The first one, basically the properties and functions of monosaccharides, and then we have to discuss the disaccharides as well. So yes, coming on to the disaccharides. Now, disaccharides, basically they are formed by joining two monosaccharides, right? And they're joined together by a bond that is called as glycosidic bond. I will be discussing this bond in detail. Okay, that is nothing but a covalent bond, guys. That is nothing but a covalent bond. I will be coming to this particular point in detail. So basically, these disaccharides, they are like divided into reducing and non-reducing disaccharide. Reducing and non-reducing. Now, when I say reducing, guys, I have already told you that they are having a free functional group. Free functional group, right? Either it can be an aldehyde group or a keto group, right? So now this free functional group is something which makes it very, very reducing, guys, right? Apart from that, when I say non-reducing disaccharides, so whenever, like, since they are made up of two monosaccharides, so one monosaccharide will have a functional group, other monosaccharide will also have a functional group, and these two functional groups are the ones which are participating in the bonding. And obviously, when they're participating in the bonding, no functional group is left free, right? All the functional group is participated in the bonding. So we have no free functional group over here. No free functional group over here. So that's make it non-reducing guys, right? So you reducing guys, you have to like mention over here. We have maltose, lactose, isomaltose, and we have lactulose also. Lactulose also. Non-reducing disaccharides, guys, remember we have sucrose, trehalose, we have already discussed this, right? So that is something you have to like keep in mind. And if we just look at the disaccharides wala part, so I've already told you maltose basically they're formed from monosaccharides. They're formed from monosaccharides, and maltose is formed from glucose plus glucose, right? We have two glucose molecules, right? Two glucose are there, and the first carbon, it is making bond with which carbon? Fourth carbon. It is making bond with carbon? Fourth carbon. So it is 1 to 4 linkage. First, okay, let me just try to show you guys over here. So we have, like, for example, this is the basic structure of glucose, and glucose is an aldehyde derivative. So it is containing this H, right? So I'm not drawing the entire structure. This is glucose. So we have another glucose also over here. We have another glucose also over here, right? Glucose again will have like these 6 carbons. Over here also, the glucose will have 6 carbons, right? Now, when the first carbon, it is reacting with the fourth carbon of another glucose, this is called as alpha 1 to 4 linkage, right? Alpha 1 to 4 linkage. Now, guys, why it is reducing? I have told you the definition of reducing. It is basically like having one free functional group. If you see the second glucose, this aldehyde group is free. It is not participating in the bonding. That's why what makes it reducing, it is the free functional group. The free functional group. They can donate electron to other molecules and can participate in the redox reaction, right? So ultimately, the isomaltose, if one, it is bonded with the sixth carbon atom, again, it is a reducing sugar because obviously in the second glucose, the like functional group which is present at the first carbon, it is free. Now, trehalose, when I say glucose to glucose, again, it makes sense over here, guys. Now, alpha 1 to 1, the first is basically linking with first. Both the like the functional groups, they are participating in the bonding. No free functional group is left. That's why it is a non-reducing sugar. That's why it is a non-reducing sugar. Again, sucrose, it is glucose and fructose. It is glucose and fructose. Lactulose, it is galactose plus glucose, right? It's a reducing sugar, beta 1 to 4, obviously. Okay. So that is the basic structure that you have guys have to remember, right? How these saccharides are formed from which monosaccharides, and what is the linkage present, okay, and whether it is reducing or non-reducing. That is something you can just take away from this chart. This chart is very, very important from the exam point of view. Again, this section has been taken from the Auricle notes, right? They have beautifully designed this. So that is like in one chart, you can just relate everything. Okay, so yes. Apart from that, guys, if we talk about the properties, also, it is again like one property we have discussed, it is reducing versus non-reducing, right? If I just want to summarize disaccharide properties, one thing that is something which you can remember, it is the reducing and non-reducing, right? Reducing and non-reducing, right? And guys, remember, they are also soluble in water. Solubility is there, right? Now, solubility may vary upon the different types of specific sugars, but yes. So yes, apart from that, you already know the sucrose, guys. It is the sweetest sugar, right? The sweetest, one of being the sweetest sugar. And we have the reducing and non-reducing part. You already know maltose and lactose are reducing. Otherwise, the rest ones, sucrose is non-reducing, right? So that is to do the glycosidic bond formation, right? Yeah. Apart from that, if we talk about the functions, I'm just talking about the functions of the various disaccharides, guys, right? So yes, the enzyme sucrase hydrolases sucrose into, okay, it's absorbed in the body, you already know. Sucrose is a significant source of dietary carbohydrates, right? And it's used as a sweetening agent in the food industry. Okay, so that is you already know. Apart from that, maltose, guys, you have to remember it is basically used as an energy source for growing embryo in seeds, and it is also used in alcohol production, right? It is also used in alcohol production. Apart from that, you also have to remember about the lactulose. Lactulose, basically, beta, it is used as a filler in most of the tablets nowadays. It is used as a filler in most of the tablets. Apart from that, in the food industry, also it plays a very, very important role, right? So yes, that is you just have to take away from this particular question. So that was regarding the question 2. Okay, the structure, properties, and functions of the monosaccharides and disaccharides. So in a 10 marks question, guys, these are something which you have to take away from these particular slides, right? So coming on to the next question, guys, for today, that is what is the glycosidic bond, guys, and explain its formation with the help of an example. Hmm. Now, guys, for the glycosidic part, you guys, I have already told you, as in like giving you an example over here. So, yeah, if we talk about, okay, let me just take one of the example over here. The glycosidic bond is basically no, the reactions which are taking place. These are called as condensation reactions. These are called as the condensation reactions. Just a moment. Okay. Hmm. Now, if we generally talk about know the maltose, okay, let us simply talk about the maltose, guys. The maltose, it is basically a disaccharide. Now maltose is basically formed from two potential glucose units, glucose and glucose, right? Glucose and glucose. Now, guys, the basic structure of glucose you guys already know. Let me just try to draw for you guys and then explain you, right? We have like two glucose, and you already know the aldehyde group, it is present at the first carbon, right? And we have like six carbons over here. We have six carbons over here, right? At the third place, the orientation of OH is over here, right? Otherwise, the orientation of OH is always on this side. Just try to balance the hydrogen as well over here, right? Only at the third carbon, guys. Only at the third carbon, you can just see the change, right? So, yes. Hmm. So yes, that is something you have to remember. Oh, okay, let me just try to correct it over here. Yeah, this is OH, OH, H and H. Now, guys, again the numbering part. Let me just try to number it. 1, 2, 3, 4, 5, 6. Again over here. 1, 2, 3, 4, 5, 6. Now, when I say glucose and glucose, basically what bonding we are actually getting, the first carbon is basically getting linked to the fourth carbon, right? So first carbon, it is getting linked to the fourth carbon. That is alpha 1 to 4 linkage. That is basically the alpha 1 to 4 linkage. The first carbon of the first glucose, it is basically bonded to the fourth carbon of the second glucose. Fourth carbon of the second glucose. Even if we just try to draw cyclic structure as well, right? So drawing for the first glucose over here, glucose and glucose, drawing for the first glucose. Okay. Now, in the cyclic structure, guys, one thing that you have to remember, it is the structure that is called as, yeah, the hemiacetal structure. So, yeah, over here, just try to remember one thing that, hmm, hmm. Now, if we just try to take the first glucose over here, so basically we are starting with the sixth carbon, right? The sixth carbon basically contains CH2OH, right? CH2OH. The sixth, it is basically linked with the fifth carbon, right? So over here, okay, let me just draw it. The fifth carbon over here, okay, we are just denoting it by like the 5 over here. Then 5 will be linked to the first one, right? 5 will be linked to the first one. In the first one, OH is over here, H is over there. Second one, you already know OH is over here, H is over here. The third one, OH is on top and H is down. Then at the fourth one, we have OH and H over here, H over here, right? So that is something you have to remember. This is the cyclic structure of the glucose. The same structure, guys, you have to draw again, just to show the linkage over here. Just to show the linkage over here. I'm not trying to draw each and entire thing again. And over here, we have the fourth carbon. We have H and OH. Now to just show the linkage, guys, remember the water molecule is removed from here, right? And obviously, guys, the structure which will be formed over here, it will be, yes, this oxygen which is left over here will be bonded like this, H and H. And this is called as maltose, right? The maltose, and this bonding, it is called as the glycosidic bond. This bonding, it is called as the glycosidic bond, right? So that is something you have to remember over here. Okay. So you can just remember from the exam point of view, it is no. The formation of the glycosidic bond basically takes place through a condensation reaction, right? Condensation reaction, also called as dehydration synthesis. We are removing the water molecule, if you can see. It is called as the dehydration synthesis. So now what are the steps, guys? You already know. It is basically taking place at the anomeric carbon of the first carbon and hydroxyl group at the fourth carbon over here that we have taken. So it's like 1 to 4 linkage, 1 to 4 linkage, right? So that is something you have to remember. Okay. So what is happening? The water molecule is getting like loss. Okay. And there will be the formation of the glycosidic bond. Okay. So yes, that is. I have taken an example. I will explain you how this goes out. Okay. Coming on to the structure and importance of disaccharides, particularly sucrose and lactose. Sucrose and lactose. Now, guys, coming on to, okay, firstly, just let me talk about the sucrose one. The sucrose. Sucrose is also called as the table sugar. Okay, it is also called as cane sugar, or it is also called as the, yeah, the table sugar, and it is formed from glucose plus fructose. Glucose plus fructose. Now, glucose, you already know the structure. We have like 6 carbons. At the first carbon, yes, the aldehyde group is there. Second, OH is over here. At the third carbon, the OH is present over here. At the other carbons, the OH is like this, right? So that is the structure of glucose. And let me draw fructose as well over here. Okay. So yes, the keto group, it is present at the second carbon. You already know, right? And yeah, the third over here will be there, right? The rest basically remains the same, right? So fructose is a keto derivative, glucose is an aldo derivative. We have already discussed in the starting. Now, what is happening, guys? So there is over here alpha 1 2 linkage, alpha 1 beta 2 linkage is there, which you can very well see the carbon at this aldehyde group. It is basically reacting with the second group. So we have alpha 1 beta 2 linkage. Basically, no free functional group is there. Both the functional group are participating in the bonding, and that is guys, it makes it non-reducing, right? Fructose, it is always in a beta form. We'll be discussing the alpha and the beta form later, right? So obviously you have to just remember over here. Apart from that, we have already discussed no lactose also. Lactose is galactose plus glucose. Galactose plus glucose. So obviously galactose is basically, okay, you just have to remember at fourth place also the OH is also on the this side. Otherwise, the same structure like glucose. Okay. And the glucose is already, the CHO, that is, yeah. So that is again the structure of the glucose and galactose. And basically what is happening? The first carbon of the galactose is basically reacting with the fourth carbon of the glucose, right? So that is again the beta 1 to 4 linkage. And obviously one free functional group, which you can see in the second part, that's make it reducing, right? So yes, that makes it reducing, right? So that is something you have to like take from here. Apart from that importance, I have already discussed in the previous slide, guys. I have already discussed that maltose. Okay, the lactose, it is used in food industries. It's used as a filler in tablet. Okay, and it's basically commonly called as the milk salt, milk sugar actually. Okay. Apart from that, sucrose, we already know it is used as a sweetener. Okay, it's used as a sweetening agent in the food industry. Source of dietary carbohydrates is there. So that is something you have to like remember. Now, the isomerism seen in carbohydrates with examples. The isomerism seen in carbohydrates with no examples. So let us just try to see all the isomerism that is like seen in carbohydrates. Firstly, you have to remember, since it's a short question, beta, just remember structural isomerism is there. Structural isomerism. And the second one that is there, that is called as the optical isomerism. Optical isomerism is there. Structural isomerism, it is also called as the stereo isomerism. It is also called as the stereo isomerism. Structural isomerism, it is also called as the stereo isomerism. Optical isomerism, okay. Structural isomerism, basically we have the same molecular formula. We have the same molecular formula and different structural formula. Different structural formula. And about the optical isomerism, guys, just try to remember it is the difference in rotation of plane polarized light. Rotation of plane polarized light, right? So when we say asymmetric carbon atoms, so basically we are talking about the carbon atom that is occupied by four different valencies, and these valencies are basically all four are different. That is called as the asymmetric carbon atom. That is basically called as the asymmetric carbon atom. Okay, so yes, optical isomerism, guys, you just have to remember ability to rotate the plane polarized light. Rotate. Okay, so yes, the property it is called as dextro rotation, right? So yes, that is like you can remember. Okay. Mhm. So you can just elaborate on these guys, right? You can just elaborate on these, the more on structural isomerism. right? The more on structural isomerism, right? For example, okay, over here you can take the example of glucose and fructose. Glucose and fructose. Basically, they have the molecular formula same, C6H12O6. The molecular formula is same, but the structure is different. The glucose is an aldehyde derivative. The fructose is a keto derivative, right? Again, stereoisomerism, guys, yes, the okay, they have the same structural formula, I would say. Okay, they have the same structural formula, but different in the arrangement of atoms in the space. Okay, they can be like further divided into enantiomers. Okay, and they can be also no divided into diasteromers, right? So enantiomers, guys, we have already talked about. They are like the mirror images of each other, and they are non-superimposable, right? They are non-superimposable, right? For example, enantiomers, we have D-glucose and L-glucose, right? So they are differing at all the chiral centers over here, right? So D-glucose, guys, it's the naturally occurring no in the biological systems, right? So diastereomerism, basically, they are not the mirror images of each other, and you already know they differ at one or more chiral centers. Okay, so that is something you can just take away from this particular slide. Reducing sugars, we have already discussed this question in detail. The reducing sugars that is having one free functional group, okay, that can participate in the bonding. And examples we have already discussed. Yes, the lactose and the maltose we have already discussed. Epimers. Now coming on to the epimers, guys. Coming on to the epimers, and you are already aware of the epimers. No. Epimers are basically the isomerism at a single carbon atom. Isomerism at a single carbon atom. Okay. Other than functional and penultimate. Right. For example, like if I just try to draw glucose. Okay. Now, glucose, you already know the basic structure of the glucose. Okay. So basically, yes. At the third carbon, the OH is over here. Now, what happens if now again, if at the fourth carbon, guys, the OH shifts to this side. Now, what will happen? Already at third carbon, the OH is here. And also on the fourth carbon, if the OH shifts here and the other structure basically remains the same. So this is nothing but the galactose. So I can basically say the galactose, it is the fourth epimer of glucose. Fourth epimer of glucose. Only at one position we are changing from glucose and we'll get a new compound that is galactose, right? So that is something you have to remember. Now, what is mutarotation and why guys this mutarotation is very, very important. Okay. Now, mutarotation is, guys, it is the process by which an alpha and beta enomer of a sugar basically they interconvert in solution. So basically they are interconverting in a solution, and what is happening, guys, what is happening, and there will be a change in the optical rotation over time. Change in optical rotation over time. Now, this phenomena occurs because many no like monosaccharides basically they exist in equilibrium between their no cyclic forms and open chain forms, right? So yeah, if we talk about the mechanism, also the cyclic forms. For example, if I'm talking about the cyclic forms, guys. Now the glucose you already know can exist as alpha D glucose. It can exist as beta D glucose. Right. Now, when I say alpha D glucose, you already know the hydroxyl group on the anomeric carbon. It is like below the plane of ring, right? And in beta carbon, basically, it is above the plane of ring. or they can also exist in the open chain form, like glucose, you already know, right? Now, what is the importance of muta-rotation? Basically, it helps in understanding the sugar behavior. Now, when I say sugar behavior, basically the properties of the sugar, right? The properties of the sugar in the biological system, how sugars are metabolized, how they are utilized in the living organism. They are basically like it helps to understand that. Secondly, you have to remember all the biochemical reactions, guys, all the biochemical reactions, all the enzymatic reactions which are involving these carbohydrates, right? So it's like important for that. For example, like enzymes that catalyze the reaction with sugars. So basically, they have preference for either the alpha form or the beta form. Okay. Apart from that, the quality control is also like very, very important, because in food science, guys, you already know the sugar industry monitoring mutual rotation can be important for ensuring the consistency and quality of the products. So that is status regarding the muta rotation, guys, that you have to remember. So we have discussed, guys, all the important questions for today. So yes, these are the various social media handles where you can be updated about the upcoming class. Thank you for your patience and for listening me out. Thank you.